You've heard already what the microbiota vault is, and I just want to really give you here my personal perspective again. I think it's very, very important that we safeguard microbial diversity on the planet. This is a very important topic, and we have to drive that over a non-profit initiative, which is global, which helps, also low and middle income countries to keep this for future generations. I really think this is important, and it's an obligation that we help each other. And this was only possible due to the visionary mind of Maria Gloria Dominguez and Martin Blaser, who really had this idea to do that. And as you've just heard and discussed, this is strongly driven also by the idea of the seed vault in Norway. And the concept is to not only include human microbes, but obviously also the One Health concept. So this could also be animal sources or food, as you're going to see in a second. So the idea is really that the local collectors remain the owners of the sample. So we cannot do anything with these samples when they are stored in a central way. We follow the Nagoya Protocol. We really follow an ethical standard that the local collectors remain the owners. So this is really like a deposit, a backup solution, and it's also very secured in the sense of quality control. So, for example, you have a secure cryogenic backup here. Now, what did we do in the pilot period? We had different work packages. We had some initial concepts. We developed SOPs. We had the interaction with the local collection groups. We also did some storage exploration. So what would be the best storage condition? And then we developed a set of metadata and also sequenced some of the samples. And I'm going to show you a few examples here, and I want to focus here on these four aspects. So the first one was, what is the best storage condition? I mean, that's an important question, because the pre-analytical way you handle the samples is very, very important. If you let the sample at room temperature, it's kind of obvious that the relationship of bacteria change between each other. And this project was strongly driven by the team of Pascal Fonesch. She's a professor at the University of Lausanne. And what we did is we took 5 stool samples from adults, children, and infants, so in total 15. We had them shipped over mail or a pickup service. And then you can see here different conditions we did. One part of the sample was directly sequenced. One part was plated and then sequenced. So there was an in-between culture step. And then we also did plating after long-term cryopreservation. And we had here the opportunity to store the sample for 1 year. And we had here 2 different cryopreservation flavors with liquid nitrogen and with a -80. And these cryopreservations here in the beginning, they were, we had here different treatment groups, so for example, snap freezing or glycerol, and a series of conditions, which you can see here. A total of 8 different conditions were compared in liquid nitrogen and in the -80. And then the plating here, this was a series of very broad agar plates to enrich the bacteria. And then everything was sequenced and compared to find out which is the best tube. What is the impact of all these conditions you basically see here? And I cannot go here too much into the detail, and the paper is submitted and currently under review, so you will can read more about it. And I just want to deliver here a few key messages. First of all, if you go and freshly sequence something, you simply have more diversity if you plate. That's not a big surprise, I would say, because obviously due to the plating, you have a selection of certain bacteria. And then you can also see if you actually go and compare the different conditions. So for example, glycerol versus, I don't know, something else, snap freezing, for example, these were more or less comparable. So we in our lab use glycerol because it's very easy to handle. But if you would decide to use sucralose, there's not a substantial difference between. Also, it does not make a huge difference if you store the samples at -80 or at liquid nitrogen. I think that's also an important message. What is quite interesting is the observation that when you have a fresh sample and a cultured sample, the difference depends a bit on what is your population. So if you look in adults, obviously adults have a very rich and diverse microbiome. And then if you go and culture, the difference is stronger compared to an infant where you have less diversity of the microbiome. Another important message is that the individual person over a year really keeps the composition of the microbiome. So you can see this here in this principal component analysis, where every single donor of this experiment has a certain color. So for example, if you take this green color here, or this purple color, or I don't know, the yellow color here, this is a single individual. And each condition we had over the time point over a year, they cluster quite nicely together with a few exceptions, but overall a very nice clustering. And that means that the microbiome is relatively stable over these years. So that's good news, but that's only 1 year. How this would look like in 10 years, I don't know. And maybe somebody should actually do this. Okay, so that was the let's say the storage, how to best store, and then how do we store the samples we have received? Because we've received from a series of local culture collectors samples, and this is a specific room in my institute, in the Institute of Medical Microbiology at the University of Zurich. It's a locked room, restricted access, and in this room we have temperature monitors for all these freezers, and we have quite a high capacity to go for about 160,000 samples at the moment, but obviously we would plan to further expand that. And we have specific emergency policies, so there's alarm systems. Even at night somebody would come in and really save the samples and transfer them to a backup freezer if necessary. We received samples from across the globe, so from Brazil, from African countries, from Asian countries. And what you can see here with the two different colors, we received human stool samples, but also fermented food samples. And you see here, for example, from Laos, we received 73 fermented food samples. So the fermented food was actually quite a challenge for us later on in the sequencing, because food is very diverse to stool, and extracting the DNA is sometimes not so easy. So we had to really establish a series of food-specific extraction protocols, and we played around with own food we bought. So for example, somebody sent us kimchi, and then we were not sure how to best extract the DNA from kimchi. And then we bought in a local store some kimchi to actually optimize the kimchi extraction protocol. The samples are managed over a professional software. This is a biobank information management software. We use here a company, dData from Switzerland, and this really allows you to perfectly track the whole process of sample storage. You have here an overview over your freezers. You can click into, you see then here lots of additional information, where the sample is coming from, contact details of the person, etc. And also the workflow was integrated. So for example, how the samples was processed, every step. And you can really see where the sample is in the process. Also, you can integrate this then later on with sequencing data. So for example, if a sample is then sequenced, the sequence data also appears here on the screen. So this was the whole workflow in an overview. So the sample, it was registered, it was stored, the DNA was extracted, and the 16S sequencing was done, and all this information came together in this biobank information system. And I think what is very important is to standardize the process in your local system. I think no matter what kind of protocol you choose at the end, really try to stick to one and not have too many changes to also make the data comparable within a project. I guess that's a quite obvious recommendation. And then the labels also, I think it's very important that the labels are resisting low temperatures. So we had here the stool samples and the food samples. What we did for the DNA extraction, we had here different protocols. For the stool samples, this was a beating protocol with tissue lyser from QIAGEN, and then we used the DNA extraction protocol from Maxwell. And you can also get all our SOPs on the website. For the food sample, we had here a Kingfisher system, and we really had to optimize here the DNA extraction. And then the last project was the 16S sequencing, and this was done together with the research group of Professor Nikola Bokulic from ETH Zurich. We followed the EMP, the Earth Microbiome Protocol, 16S sequencing with the Illumina Amplicon Protocol version 2, which was published, and we sequenced all these samples and provided then the genome sequences also to the local collectors. So that was a very nice, went smoothly. It took us a bit time, obviously, to sequence so many samples, but yeah, that also worked very nicely. We had here some controls like phage in there as a positive control, and we did all the sequencing on a MySeq system. So what are the key messages? I think the pilot project was a success. Overall, when I look back, I really was skeptical in the beginning. Does this really work? And indeed, we had some challenges to generate like all the legal frameworks to get the samples then to Switzerland. But with the contracts, etc., there was really a vice versa trust towards each other. We had samples from 7 different cohorts now, human stool and food. The storage conditions did not show substantial differences on a patient level. I think that's an important message. The governance with different partners worked well. We had lots of interactions with each other over email, and the next steps now are clear. We need to get more funding. This is absolutely necessary. And I would really like to thank here the whole pilot team, especially Maria Gloria, really such a visionary person, and also my colleagues who were a bit more on the operational side, and then also Dominic and Manuel who supported this project very much.